You're listening to the MER-Mates podcast, the marine science research podcast.
Picture this. You are at the beach with your friends, soaking up the summer sun. You know the usual hazards: sunburn, strong waves.
But what if we told you there's a microscopic danger lurking in the water? One that could send you straight to the lifeguard stand.
In this episode, we dive into the hidden world of Ostreopsis, a tiny but powerful algae making waves in the Atlantic.
But the real lifeguards in this scenario aren't on the beach, they're in the labs, where scientists are racing to understand its impact.
Join us as we explore this intriguing microalgae and hear from today's guest journey into this cutting-edge research. What are the risks? What's being done? Tune in to find out.
Welcome again to our podcast, MER-Mates. We don't want to spoil the action today, so I will keep today's introduction nice and short. We have with us the very first early career scientist, and well, he's currently enrolled in a PhD program, which he'll be telling us all about briefly. We have with us today Yago Laurenns. Welcome.
Thank you.
Hi, Yago. It's a pleasure to have you here. To start, you studied biology here in the University of the Basque Country, as I did, and then you moved to Galicia to do your master in marine biology. So to start, we would like to know what was your experience like in Galicia?
Hi, happy to be here with you both guys, and yeah, I'm glad to talk about anything you want to. So let's start with Galicia. I mean, Galicia was pretty nice experience. I was, as you said, after I finished the biology degree here in the University of the Basque Country, after a short period of working abroad and living a little bit my life, I went back to the career life and all that. And yeah, I moved to Vigo in Galicia, and I was there for a couple of years doing the masters of the marine biology masters, which was really interesting and really useful. I mean, the first year was a bit similar to what I had done in Bilbao. It was like a biology 2.0, nothing very different from what I had done here. So it was kind of disappointing. I met very nice people, very interesting researchers, but I was kind of, as I said, disappointed. And even more taking into account that it was like the COVID period, so I couldn't do everything I wanted to. Luckily, the second year was completely different. Similar to what you have here, guys, the masters got like the first year is more like theoretical. You've got more exams and all that. And then the second year is more focused on doing your master's final project, and also some like an internship in a research center or in a research group in the university. So yeah, it was great because I went to a center that belongs to the CSIC, which is the Marine Research Institute in Vigo. And I was there working with Ángel Fernández González, who is a quite important researcher in the area of cephalopods. And I was really excited because it was like my dream, because it may sound a bit delusional, but it was after I watched my Octopus Teacher documentary in Netflix.
It's quite popular.
Yeah, I know, and it's got like a like two-sided. No, it's very two-sided. Some people are like, "Come on, don't keep up talking about it. It's enough." But I really loved it, so I really wanted to give it a try and see how the octopuses and all cephalopods work and how they live. So I had the chance and I went for it, and it was a spectacular experience. I really enjoyed it. I learned a lot. I met very nice people, and yeah, and that's the thing.
Quite nice. You kind of answered some of the questions we were going to ask. But, well, in our podcast, we're learning a lot about the research that's going on here in the Basque Country. Can you maybe expand a little bit on the research you were doing with cephalopods in Galicia?
Yeah. I mean, I was I took part in like oceanographic survey, which was financed, I think, from the European Union, and there was like a lot of income coming in, and it was a big project. It had different research lines. It was focused on trying to understand the ecology of the paralarvae, which are the juveniles of the cephalopods. So focused on learning and understanding the ecology of the paralarvae of octopus in the Galician and Portuguese coasts. Apart from that, we also got any zooplankton samples and organisms that were fish larvae, copepods, and loads of different taxa. And part of my kind of try to study the ecology of the paralarvae of Ostreopsis to know how they behave. So trying to understand if during the night they prefer to stay in deeper waters, or if they prefer to go to the surface to look for more food, or what's their behavior.
You've been talking about the marine research you were doing in Galicia, but currently you're doing something very different, which we'll hope to talk about soon. But we want to talk a little bit about the process of finishing an era, a period of, for example, the master, and getting into the next one. What was that transition for you like?
It was really fast. I mean, I hadn't finished my masters. I was still waiting to present thesis in the masters. I wasn't thinking about anything particular. I was just focused on finishing that project, and after that, I didn't know what I was going to do. My idea was to try to keep or to stay in Galicia because I was really happy there. I had a couple of chances because my same director offered me the possibility to stay there and keep working with them. But the truth is that it wasn't that interesting for me because the project wasn't about cephalopods. It was more related to the parasites in the hake, in fish. So it was cool, but it wasn't my dream. So I just postponed it. I wasn't completely sure that I wanted to do that. For me, the best option would have been, at that moment, would have been to stay there working with a guy that I knew there. I was really nice, and I really got along well with him, Jorge Urcera, who works with adult octopus. So he usually studies the behavior and the ethology of adult octopus in the Thies Islands, and he tries to understand how they behave and how they move, if they prefer to stay in a place or if they prefer to move around and look for prey, for mates, for anything like that. So that was pretty cool because I could have dived, scuba dived. That was like the perfect match. But it wasn't possible at that moment because he had just started, so he didn't have the resources to offer a PhD. All in a sudden, I got a phone call from a guy, from Sergio Segane, who is my current PhD director, supervisor. We have to go back a little bit because I knew him already. He wasn't calling me for nothing. I mean, I had been working with him during the biology degree. I had done my final project with him related to phytoplankton. We got along well. That's the truth. I mean, we worked well together, and we kept in contact after I finished the degree. So I got a phone call from him, and he offered me the possibility of working with him again. the first idea wasn't to start a PhD. That wasn't within my thoughts. But I mean, the first offer was to work in a short project that was going to last for like a year or a couple of years, and yeah, it was related to monitor the ecology and the community of microalgae that is called Ostreopsis, which is a toxic microalgae. So yeah, that was the first idea. It was just a short contract to do something after I had finished my master's. But my first thought was, okay, I mean, I'm not getting into something super serious, so I still have time to think about what I want to do.
And with this, you kind of resolve the mystery of what your work is. It's about this little project, which has turned into a PhD.
Absolutely. Yeah.
What does your day-to-day look like now with the PhD?
It's nice. I mean, I can't really complain. Get along very, very well with each other. I mean, now they are friends, so it's very good. With my boss, with Sergio, is also very, 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 very good. I mean, he's very close, so he knows how to treat us. So he knows when to push and when not to, and he's not too strict. So, I mean, we've got some flexibility in terms of timetables and schedules and all that. But that doesn't mean that we don't work. I mean, it's two different concepts. I mean, to be happy at work doesn't mean that you're not working. You can be both things. So we work hard, we do a lot of things, and there's some periods that are extra busy. We've got a lot of things to do, samplings, lab work, laptop work. There's plenty of things. And other seasons that are more easy. Now my easy season is getting over. And the busy season is starting for me.
And between you and me, how is working with Sam in the same team or lab?
Well, for those who don't know, I am doing my master thesis in the same lab, and Sergio happens to be my supervisor too. It's a fantastic experience, isn't
absolutely,
it?
yeah. I mean, I'm being recorded, so what can I say? No, it's great. I mean, I think everybody that has come into the lab has been like accepted very well, and I think they can all notice the good vibes that we've got. We're happy to welcome people that are willing to work, and we've done a couple of samplings together, and yeah, he's not, he does it well. I mean, can't complain.
Well, I didn't break anything. Actually, well, I did break something on my first day, but well, I guess that happens to everyone, maybe. Yeah, it's a very interesting lab. Phytoplankton. It's not as well. It's not as people don't work with it as much as you'd expect. We're talking about carbon sinks all day and all, and yeah, people don't really know what's in the water. But we would like to know what you know about what's in the water. So we want to focus on your research and extended knowledge about Ostreopsis. Can you make us a brief summary of what's going on with Ostreopsis in the Basque Coast?
Sure. First of all, I mean, I'm not a super expert.
So you're becoming one.
Hopefully.
PhD program's all about that.
Hopefully. Yeah, it's just been a couple of years yet, so my knowledge is limited. But yeah, I'll try to give you the best perspective of how Ostreopsis is in the Basque Country. So the thing is that the first report of Ostreopsis was in 2007 in the Basque waters. It's benthic dinoflagellate, which was originally located in tropical and warm waters, so it wasn't supposed to be here. But yeah, in 2007, probably because of the global warming and different factors, we first saw it here in Biscay, and after that, there were a few years that it wasn't very important because wasn't too abundant and it wasn't causing any problems because it's a potentially toxic genus of dinoflagellates. As I said, there were a few years that it was under the radar, and suddenly in 2021 there was like an outbreak in San Sebastián and other beaches in the French part of the Basque coast, like San Juan de Luz and other places, where they were having problems with people complaining about skin irritation and different symptoms. I wasn't there. I wasn't working with Ostreopsis at that time, but the experts realized after taking some samples from the water that it might have been because of Ostreopsis, because the abundance in the samples was really, really great. So that was the conclusion, that all the problems that were lasting, I think it was in the first week of September, they were caused by Ostreopsis. So after that, Ostreopsis started to be an important factor in the Basque beaches, because at the end, when something affects humans when they are trying to just have a bathe, go for a swim in the beach, usually becomes much more important than a regular dinoflagellate or diatom or anything, any phytoplankton species or genus that is there all year round.
Mm-hmm. You mentioned maybe the most probable reason for the presence of Ostreopsis in our coast, but what conditions are needed for an Ostreopsis bloom?
It's still kind of difficult to give you the perfect conditions for Ostreopsis, because it also depends on the place, on the species, because you've got different Ostreopsis species. So here in the Basque Country, we used to have only Ostreopsis siamensis. In the last samples, we isolated a few strains of Ostreopsis, and we sent the samples for molecular analysis, and the results were saying us that we also had Ostreopsis ovata, which is supposed to be more toxic than siamensis, which is considered not to be toxic. So maybe now it's a bigger problem than before. My boss had been working with Ostreopsis for a few years back. He also reported that Ostreopsis was in Cantabria, in Santander, and different places along the Cantabrian Sea, also in Comillas and other places. They were working a lot with it. Taking into account those studies and what I have done since I started the PhD, we could say that the best conditions for Ostreopsis to grow are warm temperatures, low hydrodynamics, which means low wave action, low currents, mild winds. Also, what we've seen is that the bottom of the sea is shallow and rocky, with a lot of macroalgae, the ones that you can see on the naked eye. That usually favors the development of Ostreopsis.
Regarding this last point about macroalgae being related to the development of Ostreopsis, why is this happening?
It's because Ostreopsis, as I said, is a benthic dinoflagellate. Usually you don't find it in the water column. It's got preference for substrates, so it needs to get stuck to adhere to different substrates so it can grow and develop. So it's been observed that it usually prefers to get stuck and to live in an environment full of rocks, of macroalgae. Depends on what it has at each specific site. Usually tries to get stuck to those substrates.
So we've learned already that for a Ostreopsis bloom we need, let's say, warm waters, calm water, a hard substrate with macroalgae. This question is maybe because of my lack of knowledge, but is it the same when there is a Ostreopsis bloom and appearance of toxic toxins? I mean, you have a bloom of Ostreopsis, but does it always mean that there will be toxins in the water? Or
That's a pretty good question, because we still don't know it. It's difficult to say, because we've seen during this, because we've already been doing like a monitoring program for the last couple of years, even more. During the last 2 summers, we observed established like alert values or limits. In principle, they are for the Mediterranean. They were established by Elisa Verdalet. They mean that if you surpass the 200,000 cells per gram of macroalgae or the 30,000 cells per liter of water, so we've got like 2 different types of samples, one from the water column and one from the substrate. So if you surpass any of them, it's supposed to be problematic. But what we've observed is that it doesn't necessarily need to happen. Sometimes we recorded really important peaks, maximums of over the million cells per gram in the case of substrate samples, or above the 100,000 cells per liter in the water column. At that moment, we were in contact, in close contact with the authorities that usually manage the problems in the beaches. So every time there was a person that was feeling some symptoms that could be related to Ostreopsis, they would note it down, and as soon as they had like 6 cases or something like that, they would let us know. During that year, we only had 1 warning, and we went to that beach to take samples not the same day, but the following day, which is kind of fast. And we recorded the minimum of Ostreopsis of the whole summer period. On the other hand, with all those peaks that I was saying before, we didn't receive any warnings from the authorities. So that's quite representative of how Ostreopsis work. You don't always have a lot of toxins when there's a lot of Ostreopsis, because we think that it may depend on the growth phase. So you know that there's like an exponential phase, then when they are about to die. So we still don't know when, but maybe it's during the exponential phase when they produce more toxins, but we don't know. I mean, that's something that we haven't studied. And also it may be related to, as I said, to the species. So it's not the same to have Ostreopsis ovata or Ostreopsis siamensis. And there may be other factors that we still don't know.
So, well, it's a tricky concept, really, understanding when is Ostreopsis really danger, but we do have the standards of abundance of cells. When has there had to be consequences, as in beach closures? What areas are the ones being the most affected from your results?
Mm-hmm. During our project, we've seen that a beach that is located in San Sebastian is probably one of the hotspots in the Basque coast. It's Ondarreta, because in San Sebastian we've got like 3 main beaches: Zurriola, La Concha, and Ondarreta. La Concha and Ondarreta are almost the same beach, but they are separated like by a pier. So Ondarreta, which is next to the Monte Igueldo and all that area. So that seems to be the perfect habitat for Ostreopsis. Probably because it's got all the characteristics that I was telling you before. So it's got warm waters, it's pretty calm because it's got a pier protecting the side. And also it's shallow with a lot of rocks and macroalgae. So that's probably the main hotspot. But we've also observed that in Mutriku and Zumaya, there's been a lot of Ostreopsis too. But it's more like in specific occasions. It's not been generally. In Ondarreta, during the whole summer, from July onwards, we usually record a lot of Ostreopsis.
And coming back to the toxins, what impacts can those toxins have in the ecosystem health?
That's difficult to answer, because you mean regarding the other animals, other organisms, not us. Yeah, because there's other people, there's other guys in the water. That's important. Honestly, we don't know much about it. Just as a funny funny fact. Last year we found like a worm that was eating Ostreopsis. It was full of Ostreopsis. It had the whole system full of Ostreopsis. So there's some organisms that apparently feed on Ostreopsis. I think it's not very problematic for other organisms because I don't think the toxins affect them very much. Because I mean, even for us, the effect that Ostreopsis toxins, which are palytoxins, have, it's not very big. I mean, it's not going to kill you. I mean, no way. It's just going to cause you like a few symptoms similar to fever and skin problems, and as I said. So I guess that other organisms is not going to be very, very problematic. But still, I mean, it's something that we really have to look at and see the interactions, not only with humans, but also with the rest of organisms in the sea.
And the chemistry of the water, like let's say anoxic conditions, so do they also?
Yeah, I mean, could be, but it's true that when Ostreopsis grows a lot, it forms like some aggregations. So you can see it in the naked eye. They are microscopic, but that happens only when there's a lot of Ostreopsis. It's true that usually they accumulate in the macroalgae or other substrates. So maybe it's true that for them may be negative, because I guess that's just I'm just thinking now as you ask me, yeah? But maybe as they accumulate all around the macroalgae, for example, maybe they don't get as much light as they want to. So maybe they can't grow, or they even die, or something like that. I'm not sure. What happens is that when there's a bit of wave action or something like that, those aggregations usually release from the macroalgae or the other substrate, and they get into the water column. So we've been able to see them in the water, like just floating around. In our case, we haven't seen huge masses of these Ostreopsis aggregation. So I don't think it's going to affect in terms of anoxia or anything like that. But maybe there's somewhere that they grow crazily, and maybe you've got the surface full of these aggregations and they produce anoxia at the end of the process. I'm not sure, but not here so far.
Right, so we want to get a little bit into the sampling, you know, and
Mhm.
into the monitoring. You're recently entering your busy season, and I think it might be even busier now because you're working in collaboration with France now, I believe, with the European project.
That's correct, yeah.
So we want to know all about the sampling experience, and if you have any anecdote you would like to share with us.
Yeah, I mean, I always said that I wanted to. Because I don't know if in English you also have the same thing, but in Spanish you've got like the boot biologist, the lab biologist, or the boat biologist. So I always said that I wanted to be a boat or a boot biologist, because I really like the samplings, the field work, and all that. So it's great to be able to do it. But the truth is that it's not always as beautiful as it might look. During the summer, you may say, "Wow, it's great! You go to the beach and you get into the water and you go for a swim and all that." Yeah, kind of, but but then I mean, as I said, during summer it's super busy, so we don't really have much time to just have fun. So we just go for the samples, tita, and then go back to the lab and keep working. But honestly, sometimes we go for a swim. If it's too hot, why not? And it's always super busy, full of people, full of cars. You don't have anywhere to park your car. Then you have to carry your all the way pretty far away, and it's like anyway. And then you say, okay, so then during the winter it's easier because you've got less work and there's less people. But then we come into the weather conditions. We are living in the Basque Country, and as you may all know, the weather during winter is not very nice. I mean, the water temperature might be at around 9 degrees sometimes. So getting into the water and putting your hand, your arm into the water, taking the samples, and sometimes there's a huge wave coming in and covers you, and it's not the best experience, but it's nice. So probably the best seasons are autumn and spring, because it's like you've got a bit of both things. So you're not super busy, but still the weather conditions are pretty nice. So that's my favorite season to swim. I haven't had any funny anecdote. It's been more about problematic anecdotes. So there was once I was in Zarauz, and Zarauz is kind of known for its waves. We were doing the sampling, and it was probably, I don't know, November or something like that. So it was starting to be cold, rainy, and all that. Nowadays we are lucky because we realized that sampling during in winter with a swimsuit wasn't a good idea. So we bought a couple of suits that are water resistant. So they're pretty nice, but they're not 100% useful. So we were here in Zarauz, and we were taking the samples. So the problem about Zarauz is that you really have to pretty much a lot into the water to get the samples. It's kind of far away, even more when the tide is not very low. And I was trying to get there, and I had left all the equipment, all the material in a rock, which I thought that was safe. And all of a sudden a huge wave came in and covered me, and all the water came into my suit. I started like sinking because I had my boots full of water. No, I mean, not dangerously.
Okay.
But I was
wondering.
No, it was a big mess because I could touch. Luckily I'm pretty tall, as you can see.
Yeah.
Not the audience or the guys that are listening to this, but so I was touching the sea floor. So it wasn't a big mess. It wasn't dangerous, but it was a big mess because the same wave took all the material and started spreading it all over the beach, and had to run after it, but I couldn't run because I had left other part of the equipment in the other part of the beach, and I couldn't get wet. So it was a big mess, and I didn't know what to do. So sometimes a guy helped me. My workmate did as the same, and it didn't happen anything, but it was kind of a silly thing.
Yeah.
So it was interesting to talk and learn more about Ostreopsis, but we are coming to an end. And before we finish, some general questions we usually do. The first is, what advice or tips can you give for the people who is on the process of choosing a PhD, or even for the people who are already in a PhD?
I mean, first of all, for me, the first important thing is that before taking and making any decision, you think about it well. I mean, you have to expand your thoughts, your mind, and be able to see different options. So for me, it's important that any of you guys that are thinking about studying marine biology and maybe later on getting into a PhD, that first of all you travel. And I mean, for example, in my case, when I finished my degree, I preferred not to start the masters straight away because I wasn't completely sure what I wanted to do. So I was away for a year. I mean, you don't have to be doing nothing. You can be traveling and working. You can be improving your English or something like that. So you don't have to get into a master's, for example, just because. I mean, you have to have a reason for that. So first of all, think what you want to do. Once you have it kind of clear, you're never going to be 100% sure because it's impossible. But once you've got it kind of clear, go for it. And once you're in the masters and you realize that, okay, maybe research would be a nice idea, don't do it. No, it's just, I'm just kidding. Just think if you really want to do it, because at least it's going to be for the next 4 years, usually. And for me, even more important than the topic and the research area, which is important, of course, you have to be happy with what you are doing. And I mean, at least you have to kind of like it. You don't have to love it, I think. It doesn't necessarily need to be your dream to be working with microalgae, in my case, or anything like that. You have to like it, but for me, the most important thing is that you are happy with the people surrounding you. That your supervisor is a nice guy, a nice person, you get along well with him or her, that your workmates are nice people or that you get along well with them. For me, the atmosphere at work is probably the most important thing, because it's going to be most of your life during the following, I don't know how many years, it's going to be that. So I would recommend to give even more importance to that factor than the other one.
And regarding the joke you did about not involving in research, do you consider that the work as a researcher is well valued or appreciated?
Honestly, in Spain, I don't think so. It sounds kind of, I'm doing it and I'm complaining about it, but it's the truth. I mean, I'm kind of lucky because I'm living in Bilbao, which is my birthplace. So I'm living with my family. So I don't have to pay the rent of an apartment. I pay part of the food and general things, but it's not the full thing. So I'm kind of lucky, and I'm being able to save money and not spend the whole salary in just living. But that's not the general thing. I mean, usually people go to other universities, other centers in different cities, different places all around Spain, and they have to use most of their salary to pay for the life. I mean, they're not able to save, and it's 4 years. I mean, most people finish the PhD when they are about 30 years, maybe a bit less, 28, 27, but some people 30 something. It's too late to not to have anything in your bank account, so you can't even think about buying a house or moving out. It's sad, but I mean, it's true that at least we are doing what we like, but I think that they should give more importance to what we are doing. It's during the COVID, it was nice because people started to realize how important science might be, because they were saving a lot of lives, and there were a lot of biologists working with the vaccines and all that, and they were supporting us. Sadly, after the COVID thing has finished, now they have forgotten us. No, they don't keep thinking about us, and the conditions haven't really improved a lot. I mean, it's true that during the last years they've improved, but not accordingly to the rest of the sectors. So yeah, it's sad. Here in Spain, it's kind of sad. Probably in other countries in Europe, in the United States and other places, it's probably easier, and they're more recognized, and their work and what they do, and not only with the salary, but also publicly, the public opinion. But hopefully it will change soon.
Hopefully before I start doing a PhD, because I can relate to not having any savings in my bank account, and it's not a nice experience. I can agree with that.
Absolutely.
Very nice advice. Thank you, Yago, for that. We're reaching, as well, we've said it before, I'm not going to repeat it. Before we end, we would like to know a recommendation you can give our audience of a book, a scientific paper. I know this sounds really, posh. We don't even read that often, Shabby and I, especially Shabby. So if you want to recommend a film, you're allowed to recommend a film, but what would you like to tell our audience to get into?
I mean, I'm probably very similar to you guys. I mean, I'm not a big science reader. I'm not reading science books every day. I'm not a science. I'm not mad about that. So there's this book that I read a few years back, and it was a recommendation of one of my professors, And I was okay. Let's give it a try. It was Wonderful Life by Stephen Jay Gould. It was about the Burgess Shale, like the archaeological sites in Canada that were really, really important. And I started reading and I couldn't finish. It was. I mean, for me, it was too boring. It was too. I don't know. So, I mean, there's a book that it's not very scientific, but it's kind of it's about marine biology, about marine organisms, and I think it's a very nice book to recommend to someone that might be interested in this. I mean, not for like very scientific people, but it's all what they say is correct, and all the information they give is good and is accurate. So it's a good one. It's The Memory of the Fish. It's La Memoria de los Peces. I read it in Spanish. I don't know if you've got the opportunity of reading it in English, but it was very nice and I really enjoyed it. It tells you about different curiosities and adaptations of different organisms in the sea life, and it's very, very cool because I read it, I mean, a few months ago, so it's not like I read it when I was 14, so I've got an idea of how marine biology is, and I really enjoyed it, and I've recommended it to a few, my family and a few friends, and they really enjoyed it too.
There you go. Well, we know that sometimes recommendations don't work, apparently, but make sure you have a check at that. Well, we've reached the end of the episode. We can only say thank you very much for being here with us. Thanks for putting maybe a little bit of our feet back on Earth when we think about research. And thank you also for teaching us all about phytoplankton, toxic phytoplankton, in the context of the Bay of Biscay.
Yeah, thank you, Yago.
No, thank you guys. It's been a pleasure.
And anyways, well, we'll see you in the next episode, and thank you for listening.